Inhibitors of replication, transcription, and translation. In this video, we are going to discuss about replication, transcription, translation, and their inhibitors. So what are inhibitors? Inhibitors are nothing but nucleic acid inhibitor, and it is a type of antibacterial. Their main function is to stop the production of nucleic acid, and they fall under two major classes, namely DNA inhibitors and RNA inhibitors. DNA inhibitors act as topoisomerase inhibitors. They also act on anaerobic bacteria. They incorporate metabolites in the two DNA strands, which causes more breakage of the DNA strands. RNA inhibitors act upon RNA polymerase that are DNA dependent. Replication. When a double-stranded DNA molecule copies itself to produce two identical DNA molecules, then this process is known as DNA replication. This is a very important process as the two new daughter cells formed after cell division carries the same genetic information as that of the parent cells. And this happens due to the division of the DNA molecules. The replication process of DNA starts at specific site known as origin of replication, and this is a region where DNA double helix starts to unwind. A small segment of the RNA known as a primer is synthesized and then forms a new start for the DNA synthesis. The cells have the ability to proofread the newly synthesized DNA. Once the DNA replication is done, the cells can then divide into two new cells. Some of the major inhibitors of replication are quinolones. These are highly active against wide range of gram-negative and gram-positive bacteria. Since these have good tissue penetration, they are advantageous for deep-seated infections. Quinolones are mainly used for the treatment of diseases like sexually transmitted ones, UTIs, respiratory tract infections, bacterial enteric infection, bacterial endocarditis and meningitis, skin and soft tissue infections. Afidocolin block the cell cycle at early S phase. They are inhibitors of eukaryotic nuclear DNA replication. 5-fluorouracil prevents the biosynthesis of thymidine during DNA synthesis, and hence it is a pyrimidine antimetabolite. Emetine dihydrochloride blocks the protein synthesis irreversibly by inhibiting the movement of ribosome along the mRNA. It can also inhibit the DNA replication in early S phase. Few other inhibitors of replication are ganciclovir, lomifloxacin, mitomycin C, penciclovir, and trifluoroperazine. Transcription. This is a process by which the information that are present in the DNA are copied into an mRNA molecule. This copied information will further act as a template for translation by which proteins are synthesized. Transcription inhibition can be done either by slowing down RNA elongation or covalent or non-covalent modification of RNA polymerase. Some of the inhibitors of transcription are rifampicin. It is an inhibitor that binds with the beta subunit of prokaryotic RNA polymerase. It inhibits prokaryotic transcription initiation. Since it binds only to bacterial RNA polymerase, therefore it is a powerful drug for the treatment of bacterial infections. They are mainly used for the treatment of tuberculosis and leprosy. Actinomycin D. It is an antibiotic which inhibits transcription in both bacteria and eukaryotes. The planar portion of the actinomycin D is inserted into the double helical DNA which is present between the GC base pair. This leads to the deformation of the DNA. Alpha-amanitin. This is another major inhibitor of transcription. It is a cyclic peptide of 8 amino acid, and it selectively inhibits only the RNA polymerase 2 and 3. It interacts with the bridge helix in polymerase 2. This then interferes with the translocation of RNA and DNA, which is needed to empty the site for the next round of RNA synthesis. This leads to slowing down of the rate of elongation. Next, we have translation. In the cytoplasm or endoplasmic reticulum, protein synthesis after transcription of DNA to RNA in the cell's nucleus takes place by the action of ribosomes. This process is known as translation, and the entire process is called gene expression. Translation takes place in 3 steps: initiation, elongation, and termination, just like replication and transcription. Inhibitors of translation. Some of the major inhibitors of translation are chloramphenicol, erythromycin, streptomycin, neomycin, and netlimycin. Chloramphenicol blocks the elongation and peptide bond formation. Erythromycin, on the other hand, blocks formation of initiation complex. Streptomycin is mainly used for the treatment of plague and multiple drug-resistant tuberculosis. Neomycin is used mainly in the empirical treatment of superficial infections of skin. And netlimycin is just reserved for bacteria that are resistant to other aminoglycosides. Some other major inhibitors of translation are gentamicin, amikacin, and tobramycin, which are used in serious infections that are caused due to aerobic gram-negative bacilli, including Pseudomonas aeruginosa. These are used in the treatment of serious infections and bacterial endocarditis. These are the references.